Welcome to Stenzel Health Solutions, Foundations of Cellular Health. This video will show you the most important aspects to a healthy, functioning cell, a foundation for describing to patients what a healthy cell looks like, and, as we will later show you, where failures occur that will lead to cellular destruction. To start off, it is worth noting the power of deoxyribonucleic acid, also known as DNA. DNA is the knowledge or blueprint of the cell required for all cellular life. It plays a very important role in creating important enzymes to carry out important functions like glycolysis. A way for DNA to express its functions within its cells is partnering with what are called sirtuins or SIRTs. SIRTs are proteins that play an extremely important role in gene expression, or the ability for a cell to create proteins needed to appropriately react to its environment. It's also involved in DNA repair, metabolism, and longevity. It's important to understand that SIRTs require the partnership of intracellular NAD+ to function appropriately. NAD+ is known as nicotinamide adenine dinucleotide, which is a derivative from vitamin B3. NAD+ is involved in over 400 chemical reactions and is important in carrying out the following functions: DNA repair, DNA replication, mitosis, also known as cellular replication, glycolysis, and other energy-producing processes. Because NAD+ is a coenzyme, it participates in redox reactions. Moving forward, it's important to follow the readily available give and take functions of NAD+. It's also worth noting that NAD+ can be located in both the extracellular and intracellular portions of the cell. But the vast majority and most important NAD+ that we will focus on is the intracellular NAD+. There are two measurement types found in research. One is NAD+ to NADH ratios, which should be at about 500 to 2000 NAD+ to 1 NADH. Another healthy measure, which is how this program will suggest measuring NAD+, is maintaining 40 to 100 micromoles of intracellular NAD+. Of all intracellular NAD+, about 30 to 60% is located within the cytoplasm of the cell. The other 40 to 70% is found within the mitochondria of the cell. Now that we have established normal levels of NAD+ and already discussed the importance of its interactions with SIRTs, let's see how NAD+ works with the important energy-producing pathways of the cell. NAD+ is consumed in both glycolysis and the Krebs cycle. Glycolysis takes place within the cytoplasm of the cell, and the Krebs cycle takes place on the inside of the mitochondria. As we see in the picture, NAD+ is being consumed to carry out the functions of SIRTs. As a reminder, the enzymes used in glycolysis are created by DNA, emphasizing on the importance of keeping DNA healthy. Furthermore, while we appreciate the enzymes used in glycolysis, we will only be recognizing the formation of G3P and pyruvate moving forward. We also see how the free NAD+ is also consumed to break down glucose, with an ultimate goal of creating pyruvate. Now we will see how NAD+ is used within the mitochondria. The important things to note about the mitochondria is it is the powerhouse of the cell. It produces the vast majority of our cellular energy while also consuming oxygen to carry out its functions. As a reminder, pyruvate is the beginning ingredient for the Krebs cycle, which is where the other 40 to 70% of NAD+ is located. Pyruvate production is incredibly important as it has the ability to cross the mitochondrial membrane and into the matrix of the mitochondria. There are three important features that allow for the mitochondria to function. Not only is there an outer wall of the mitochondria, but also a very important inner membrane. This inner membrane allows for the separation of spaces called the matrix and the inner membrane space. The next important mechanism within the inner membrane is the electron transport chain. As the Krebs cycle utilizes NAD+ and enzymes to break down pyruvate, NAD+ helps with the movement of electrons to the electron transport chain. This is where the electron transport chain will take an electron from NADH and force the electron across the inner membrane barrier. Because of the force electrons being pushed across the inner membrane via the electron transport chain, energy potential is created. This energy potential allows for ATP synthase or ATP creation to occur. So let's quickly review the 3 steps for the energy production to occur within the mitochondria. Step 1 is pyruvate enters the matrix of the mitochondria and starts the Krebs cycle. This leads to NAD+ carrying electrons by converting it to NADH. Step 2 is the electrons from NADH are brought across the electron transport chain, creating an energy potential. Finally, for step 3, the energy potential is utilized to carry out the ATP synthase process. This is the end of the Foundations to Cellular Health video.